listen a little bit about the drone work that we're doing. If you read the trade magazines, you can see drones are very popular these days. And this particular project, we wanted to look at, is there any potential for using drones to manage irrigation in berry crops? So this work was funded by a specialty crop block grant from WSDA. Collaborators on this project include Lisa DeVetter, Chris Benedict, Lav Cott, and Troy Peters, all from WSU, and a lot of grower cooperators that I've listed here. That's helped out with this project. And without them, we really couldn't have done this work, because all this work has been done in commercial fields. So determine irrigation requirements. So there's a few ways you can do it. Maybe one of the most common ways is just to look at what's the moisture in your field. So you could have sensors out there, or you just might use a field method to see how wet the field is and decide when to irrigate. And that's great. I mean, it does give you some information. Problem is, there could be a lot of variability in your field. And the other problem with it is it really doesn't tell you how much water you need to put on your fields. So over the years, since the 1970s, there's been some development using weather data to estimate your crop water requirements. You've probably gone to this Ag WeatherNet site, and on this site, one thing that you can get are estimates of what your crop water requirements are. And the way that works is basically there's weather stations throughout the Pacific Northwest. You kind of take the one that's closest to your field site, and from this, what they do is they estimate the potential evapotranspiration. And what that means, that's the potential amount of water that is lost by transpiration from plants and evaporation from soil surface in a reference field. So something like grass or alfalfa. And this is based on many, many years of good research, where they got good estimates of, based on weather conditions, what a grass field or what an alfalfa field, how much it would use, depending on how hot it is, how windy it is, and so forth. Now, of course, a blueberry plant or a raspberry plant is going to use a different amount of water than a grass field or an alfalfa field. So what we do is we have what's called crop coefficients to convert the potential evapotranspiration to a value that's representative of your crop of interest, say blueberries or raspberries. And that crop coefficient curve will change over the season depending on how big that plant is and so forth. So for example, I have one here is a crop coefficient curve for blueberries, and you can see as the leaves develop on the plant, the relative amount of water that it's using relative to an alfalfa field will increase to just slightly above a 1-to-1 relationship once we reach the first blue fruit stage, or basically full canopy development. Now these crop coefficient curves, they can work quite well. However, for berry crops, they're theoretical. Nobody's actually done the measurements. We're doing some work now to develop some actual crop coefficient curves for blackberry and blueberry. So we'll have some of this information. However, it also assumes that every field is basically going to have the same crop coefficient. But of course, you got a young planting, it's going to be very different than a mature planting. Some cultivars made different than others. So there's a lot of advantages and drawbacks to these methods. So the goal of this project was basically to use drones to estimate the water requirements and irrigation needs in our fields. And the idea is that we'd be able to fly a field and get estimates of what the water requirements are on a site-specific basis. So regardless of what your cultivar is or what stage of development is, we can get good estimates of what the water needs are in that field. So we have this drone from ATI AgBot, which is a really small company that we went with. The reason why we went with this is this is basically one that's you could change, and it's great for research purposes. It's not something I'd recommend for a commercial use, but for our purposes, it works great. Has a flight time of about 26 minutes, but we usually try to keep that from 20 minutes because if your battery dies, drops out of the sky and you lose your drone. So usually about a 20-minute flight is what we do. Top speed's 38 miles per hour. And based on FAA regulations, we stay between 50 and 400 feet with this particular drone. Now on our drone, we have two different types of camera. We have a multispectral module and we have an infrared module. So the way the multispectral module works, it's multispectral, meaning it has different, it measures different spectrum with different camera lenses. This particular one has 5 different lenses, so it measures blue, green, and red wavelengths. It also measures what's called the red edge and near infrared. And if you look at this graph, you can see a difference on the relative amount of reflectance you get off of a healthy plant versus a stressed plant. So if you look at the green, for example, a stressed plant will reflect more green than a healthy plant because a healthy plant's absorbing more of that green light. But if you go to the near infrared, it's the opposite. The healthy plant is reflecting more of that near infrared. So basically, because of these differences, we can get these indices to kind of see what's happening in the field. One of the most common indices is what's called Ndvi, or Normalized Difference Vegetation Index. This was an indice that was developed years ago by NASA. Basically, what they used it for was to just get a global estimate of how much vegetation there is on the planet. So for example, like I said, a healthy plant is going to absorb more red light, for example, and reflect more of the near infrared. So if you take the difference between near infrared and red that it's reflecting, divided by the sum to normalize it, that gives you a higher value in a healthy plant than you get in an unhealthy plant. So what does that all mean? Well, it basically, what it allows you to do is it gives you different visualizations of what's happening in the field. So here's an example of some indices that we can get with software when we fly a field. So RGB would be just almost like visible light. So it's the red, green, and blue. It's the visible lights, visible wavelengths that we could see. Next to that is NDVI, or you could do other types of indices like the chlorophyll indices or the berry or health indices it's called. And as I mentioned, we also have the thermal camera, which is just measuring the near infrared, and that's related to the temperature. And I'll talk a little bit about that towards the end of my presentation. So with this, we get layers of information from a field that we could put together to kind of assess what's happening. So to capture an image, it's all programmed. We have this software that you basically, you pull up a Google Earth image of your field, you trace out the field that you want to fly. Then the software calculates basically how many paths it needs to take to collect those images, to get it within 20 minutes and how high it has to fly and so forth. It does it automatically. So my technician, Scott, here in the field, he basically sets that up. He manually puts it up in the air, then puts it to autopilot, takes the images, and then brings it back and gets that information. So here's an example of a flight path that you might do. So all he did was basically draw a rectangle around that field, and the software automatically figures out that flight path. And from that, we see here's an example where the drone flew at 73 meters altitude at a speed of 24 kilometers per hour in a time of 22 minutes, and it collected all that information on an 11-hectare field, so approximately a 25-acre field. On the left is the red, green, blue, so the visible image. On the right is an NDVI. Here's another example. This was a Wakefield raspberry field. Here it was a lower altitude, 45 meters, 4-hectare field, about 10 acres. And you can see that the resolution is quite good. So I'm going to blow that up here. So in that section, we see a section here where there's some early signs of root rot. We take a look at that, and we can clearly see in both the RGB and the NDVI the presence of this Phytophthora. Now, if you walk that field, you can't see it, but looking at it with the drone, it's pretty obvious. Some things we can do is we can calculate the percent cover when we look at these images. We can use that information to basically then calculate what are the water requirements. So one of the fields that we compared was we had these two sets of fields that were side by side. We had a Meeker field and we had a Wakefield field. And if you know raspberries, you know Wakefield's a lot more vigorous than Meeker. So we wanted to know what are the water requirements between Meeker and Wakefield. So here's the Wakefield field on the same day. And obviously you can see there's a lot more canopy in the Wakefield than in the Meekers. And if we then fly that field, we get this sort of information. We get the canopy cover. And what you can see is, of course, we got a lot more canopy cover with the Wakefield over the course of the growing season than in the Meeker. We know from other research on many different crops that there's a good linear correlation, fortunately, between canopy cover and crop coefficient. So now what we can do is we can convert that information that canopy cover to an actual crop coefficient. Now, as I said, we're putting in weighing lysimeters to actually get crop coefficients. Once we have that, we'll get even a better relationship here between canopy cover and crop coefficient. But right now, this seems to be a pretty good way to go. So for example, if we look at the meeker with 40% cover, well, the crop coefficient in that case is about 0.68. So in other words, at 40% cover or maximum cover, it's using about 68% of the water that an alfalfa field would use. Let's compare that to Wakefield, and that's using almost a crop coefficient of 1. So it's using almost as much water as an alfalfa field would use. So if we draw that out over the course of the growing season, we got our crop coefficient curves now for Meeker and for Wakefield. And you can see there's quite a bit of difference. This is telling us that basically we're using a lot more water in a Wakefield field than a Meeker field. So if we're just scheduling irrigations based on what's coming off aggregate, that's not necessarily going to tell us the correct amount of water for one or both of these cultivars. So here, for example, raspberry water use is equal to alfalfa in the Wakefield, but it's only about 60% in the Meeker field. And if we look at that theoretical crop coefficient curve, you can see it looks pretty similar. You can see that in Wakefield and Meeker. You can see how it increases from bud break to full cover, and then over to dormancy, it declines a little bit. So Wakefield appears to be pretty close to the theoretical crop coefficient curves that we're using for raspberries. However, Meeker, not quite the same. So the idea is that what we're trying to do now is to try to simplify this, because growers aren't going to go out with a drone, fly the field, do all this math and so forth. Consultants might be able to do something like this, but what we're hoping to do is to somehow to streamline this process so that we can have this information go to a mobile irrigation scheduler that was developed by WSU to get that information, to get site-specific estimates of the water requirements in your fields. As I said, we're also doing this in Blueberry. Here's a Draper blueberry field. Here's some Ndvi images over the course of the growing season. You can see it gets greener as you go from bud break to late green stage to first blue, and then it starts getting lighter as you go from harvest to pre-senescence in September. So again, if we look at these relationships, we have a good relationship between the canopy cover and the Ndvi, as well as your crop coefficient curve. And if we compare that to what alfalfa would use, we could see in this particular Draper field, which was a nice healthy field of Draper that had good canopy development, we were at over what an alfalfa field would be using. And again, if we look at that to the theoretical field, it's similar. One thing you do see is that spike at first blue. And we know from research on blueberries that we do get a spike in crop coefficients or the amount of water that blueberries use when the fruit are expanding. Because what happens is as the fruit starts to expand and get bigger, the canopy starts to flatten out and it's intercepting more light. And when it does that, basically what happens is it's using more water. Once you harvest that fruit, the canopy goes back up, the amount of light it's intercepting goes down, and the water use goes down. So we do tend to see that spike. So I wasn't surprised at all by this relationship that we found. As I said, there's other indices that we can use to look at the field. So for example, in that same field, we looked at what's called the triangular greenness index. It's basically how green are those plants in the field. And we noticed that, there are some spots in the field that appear greener than others. You really don't see that on the other image, the RGB image, but you can see it very well when you look at these other indices. So it allows you to ask questions. Why is that happening? Here's a good example. This was another field of Draper. On one side, we have the RGB image. On the other side, we have Ndvi. These 4 rows, they all look pretty uniform in both of these images. However, if we look at what's called a green difference vegetation index, we now see that the 2 rows, to my right, your left, are greener than the other 2 rows. So by looking at this green vegetation index, which is basically near infrared minus the green wavelengths, This index was designed basically to predict the nitrogen requirements of corn. So we looked at this as a possibility in blueberries, and what we found after we went through to try to understand why were these two rows different than the other two rows, we took some leaf samples, analyzed it for nitrogen. Well, we found those rows that were showing up greener in this particular image had adequate nitrogen levels, whereas the other two rows were actually deficient in nitrogen. So this is now telling us that not only can we use this for irrigation management, but we might be able to use it for nutrient management as well. We're also looking at the possibility with Tom Purebolt's group, with Jason Meyer and so forth, that we are looking at the possibilities of looking at for diseases and so forth. So there's a lot of applications that we can do with these drones. So here, for example, is a Wakefield raspberry field. We have the RGBI. On the right is another Ndvi. In this case, I didn't do it in color. I just did it in black and white. And you can clearly see a strip across the field where we don't have as much canopy development. And that's because there's root rot in that section of the field. Well, what we did is, what you could do with this imagery is you can also develop an elevation map in your field. And when we did this, you could see that basically that section of the field where there's a lot of root rot was actually a lower section of the field. So what happens is that creek next to the field, during the wintertime, it rises, sometimes floods that field. Consequently, you're getting root rot. Now, had the grower flown that field before he planted the raspberries, perhaps he could have done something about that. He could have put more drain tiles or something, some way to correct that issue so that he wouldn't have that flooding going on. So there's some potential here that you can use this as a tool to reduce some problems that would happen otherwise. So the other camera that we have is a thermal imaging camera or an infrared camera. And the way this works is plants transpire. When they transpire, they lose water. When they lose water, just like us, when we sweat, it cools them down. Well, with the infrared, we can basically pick up the temperature of the leaves. And what happens is when a plant is stressed, the stomates on the leaves will close. And when those stomates close, it doesn't transpire as much, and the temperature of the canopy goes up. So we can then detect that with a thermal imaging camera. So here's an example of where the thermal imaging could be used on a relatively simple basis. So here on the left, on one side, we have the Ndvi image. On the other side, we have the thermal image. Now we can see that there was a difference in the canopy development in the Ndvi, but we don't know why. Well, if we looked at the thermal imaging, what we saw is that those rows that are higher temperature are essentially more stressed. So what happened is that those blocks were actually, there was something wrong with the irrigation where they did something wrong when they must have fertilized wrong or something where they caused precipitates. But for whatever reason, those rows where we have the least amount of growth, the higher temperature, they're not getting enough irrigation because the irrigation lines were plugged up. So we're able to use this basically to see these sorts of things. So this is just on the simplest basis. So basically you can see as you go from mild to severe stress, how that canopy goes from a cooler blue color to a warmer pink or yellow color. So what we're trying to do is we're trying to develop an index where we could fly a field and get the water status of the field. So what that would allow us to do is basically get an idea of exactly how stressed different parts of the field are. So for example, we have a study here where we had 3 different irrigation levels in a field of blue crop where we either, for 2-week period, either pre-harvest or post-harvest, we either fully irrigated the plants, or we reduced irrigation down to 30% for a period of about 10 to 15 days, or we didn't apply any irrigation at all during that period. And of course, you get a good relationship when we do this. If we look at the water status of the plants using a pressure bomb, which measures stem water potentials, you can see that as the plants got less irrigation, that stem water potential went down, and of course, the amount of water in the soil went down. So there's a nice relationship there. We look at the actual yields. Well, what happened is there was no difference actually when we withheld irrigation when we reduced irrigation to 30%, but we had a slight reduction in yield as well as a slight reduction in berry weight when we didn't apply irrigation. No surprise. That wasn't the point of that. The point of what we're trying to do here is to develop that relationship from what we see in the thermal images and what that actually means in terms of the water status of the plants. And so, for example, we could see that with the thermal imaging, we can measure that with full irrigation, the canopy was at 86 degrees Fahrenheit. At 30% irrigation, it dropped down to 87. It increased to 87 degrees Fahrenheit. And then with no irrigation, that canopy was 89 degrees Fahrenheit. So if we look at that relationship between the stem water potential and the canopy temperature, you can see that basically there's a relationship. So what we're doing is we're taking lots of measurements like this, taking stem water potential measurements and relating that to the canopy temperature so that we can develop basically a map such as this called the Crop's Water Stress Index, so that we can map out the amount of water stress that's in the field. So in this particular field, we could see the parts of the field that are well irrigated versus those that are not very well irrigated. What it will also allow us to do is that we can develop relationships between the amount of crop water stress and leaf water potentials to give us an index basically of the amount of water stress that's actually happening in the plants. So this is some of the things that we're working on in both blueberries and raspberries. So clearly there's a lot of potential here for using these drones for a lot of purposes in agriculture, including management for irrigation. So unlike using soil sensors where you're very limited to how many sensors you have and where you are in the field, or using agromet to kind of estimate your water use, here we can use a drone to get very site-specific on a block-by-block basis what the irrigation needs are. But we can also use it to assess the amount of diseases you have in the field. Perhaps we can use it even for nutrient management. The possibilities, there are a lot at this point. So the other purpose that we think that this could provide is a good tool for avoiding water limitations in your field. Flying the fields, looking at it and identifying sections of the fields where you see water stress. We're going to start putting numbers on that, but basically telling you if you're under irrigating or not. So one last thing that I'm going to talk about here, I'm just going to show you two slides of some work that was related to this project that has to do with pole strip irrigation in raspberries. We did it in blueberries too. Didn't see the response, but any response in that case. But this work in raspberries, I just wanted to briefly show it to you because it's pretty cool. It's pretty interesting. So what we did is we had a field of wake field that we irrigated by one of two methods. One was conventional irrigation, where the growers basically irrigated every 2 or 3 days, or whatever their regular irrigation schedule. And we did another treatment called pole strip irrigation. Now this is a study that was basically grower driven, something that they were interested in us looking at. So we designed a study to look at that. And our treatment plots are actually not tiny little plots, but they're entire rows. So each treatment was applied to an entire row of raspberries, and it was replicated 4 times. So for pulsing, what we did, rather than just irrigating once every 2, 3, or 4 days, whatever they applied during those say 2 or 3 days, we also applied that same amount of water, but rather than applying it all at once, we split it up into pulses. So basically what we did is that we applied 30 minutes of water every hour and a half until we got the amount of water on that we wanted. And the idea is what that allows us to do when you do that, in theory, is that it allows the water not to move downwards as much, but to move laterally. It also will do the same thing with nutrients. So the theory is that if you pulse strip, that you'll have less deep percolation and more efficient use of your water and nutrients. So we've been conducting this study for 2 years now. And first year, we didn't see a lot of difference, but didn't really expect it at that point, because by the time we got the treatments initiated, they already were close to the beginning of harvest. But this last summer, what we started to see was some differences between yield with pulse strip versus your standard irrigation. So here we have on the top figure, we have the yield per acre on every harvest date. And what you can see that initially there was no difference between the yield between the pulsed and the standard. But as we started getting later and later into the harvest season, we start to see slightly higher yields, but significantly higher yields with the pulse drip than with the standard irrigation. And that we also saw some differences. We didn't take a lot of measurements this particular year, but we also saw an increase in berry weight with the pulse drip. So if we look on average, what we found was with pulsed, we got 1,100 pounds more fruit per acre with pulsing than with standard drip irrigation. So there does seem to be some potential here. So we're going to follow this another year. So we're pretty excited about this. And again, the average berry weight was slightly higher on average, which would account for that differences. There was also a little bit of differences in the other fruit quality characteristics. We looked at soluble solids, titratable acidity, and the sugar acid ratio. And basically there was a slight difference, slightly less soluble solids, slightly more acidity with the pulse than with the standard. What I think could be happening, just a guess here, is that perhaps we're getting bigger berries, and when we go through with the harvester, we're knocking them off maybe a little less ripe than with the standard irrigation. I don't know. But for whatever reason, there was a slight difference. Whether we can actually taste that difference, I doubt it, but there was a significant effect. So I guess I'll stop there and ask if there's any questions.